Hi, everybody. So I'm Dr. Sanjay Bidichandani. I'm an FA researcher here at OU. And I just wanted to thank everybody for being here and for your support and generosity. Honestly, this makes a researcher feel really good that the community is behind us and we're all on the same page and we're all trying to get to the same thing, which is the cure for this devastating disease called FA. I'm here to introduce another FA researcher who's a superstar. He's a professor of neurology at UT Southwestern, and he'll just be coming up in a second. His name is Dr. Marek Napierala, and he does a lot of things for FA, and I've known him for at least 20 years, and he's, like I said, a superstar researcher. But I just wanted to give you one sort of angle on the kinds of things that are necessary for research in FA to happen. So obviously, people with FA are seen in the clinic, and we collect blood samples, but in research labs, you need to work on people's cells and people's DNA, on people's genes, to be able to come up with a cure for FA. And Dr. Napierala, in addition to the research that he does, he is one of the world's biggest repositories for cells from people with FA. So somebody in Europe wants to work on people with FA, he will ship them via FedEx cells from people who have FA. And when I say cells, now we obviously know that people with FA have a neurological condition and they have a heart condition. He will even send them cells that are neurons, like brain cells and heart cells from people with FA. So he's got the technology to actually make these cells from the cells that you get in the clinic, and he'll make these nerve cells and heart cells and send them around the world. And this just gives you another angle on what it takes to actually come up with a cure. It's not just clinical trials. There are a lot of things that have to be tried in the lab, and then they get into people. And so I'm going to introduce Dr. Marek Napierala.
Well, thank you so much. And it is my first time, our first time at Soirée. And as a good scientist, I do have a very short PowerPoint presentation. So if you look at the slides. So yes, we have a lab currently in Dallas at the University of Texas Southwestern Medical Center. But the accent is not from Texas. I was born, raised, and educated in Poland. Okay, so just to clarify that. And we have a laboratory which is focused exclusively on FA. We only work with FA. We have been doing it for way more than 15 years. And I think on the next slide, we have kind of an overview of different projects, a picture of the lab, and this is a different projects which we are engaged in. We look for not only a cure, but we look for molecular basis of the disease, which then can lead to finding different modes of therapies. We also work with specific Friedreich samples patients. Not all Friedreichs are the same. So we work with samples with patients which we called point mutation. Those are rare patients, but they exist. We worked on developing specific assays which can help developing a cure. So on the next slide, I wanted to point out what Dr. Bidichandani, Sanjay pointed out, that this is a human disease, yes? When we're discovering a drug or a cure, we cannot really work initially in humans, yes? We have to work in something which we called model systems. What are model systems? And you can see those on those slides. This is bacteria, E. coli. It's a very common model system we work in the lab. Actually, some of the basic mechanisms of Friedreich ataxia were discovered in E. coli, in bacteria. Yeast, common yeast, are very good scientific model. We heard for sure about Drosophila, fruit fly, a good model. Mouse model, everybody heard about it. But what my lab specializes in is human cells, human cellular models. So those are cells which are actually taken from FA patients, their family members, and unaffected controls. And on the next slide, it's just an introduction. We established that a few years ago. It's called the Friedreich Ataxia Cell Line Repository. And on the next slide, it outlines how the process works. So we collaborate mostly with Dr. David Lynch, which probably most of you know, in Children's Hospital of Philadelphia. When he sees a patient with FA, the patient donates a piece of a skin. This piece of a skin from the arm is sent via FedEx overnight to our laboratory. And we develop from this piece of skin, we develop cells called skin fibroblasts. And you can say, well, what skin has to do, and this actually, the picture shows those the process of development. It takes about 2 months to get those cells. What skin has to do with FA? Well, it can. So on the next slide, it's a representation. This shows what we actually have in the repository. Currently in the repository, we have cells from the skin of about 80 Friedreich's patients. We also have a different type of cells from these patients, and I'm going to tell you about it on the next slide. Actually, on the next slide, we have also, okay, so let me divert for a second. So what we can do with those skin cells? The skin cells can serve to generate cells called induced pluripotent stem cells. What are those induced pluripotent stem cells? These are the cells which we can use to generate cells which are affected in Friedreich. So we take skin, we make those induced pluripotent stem cells, which can be then differentiated into different types of cells. So from those induced pluripotent stem cells, we can make neuronal cells, so the cells which are in our brain, affected in Friedreich's. We can make from those cells called cardiomyocytes, cardiac cells, which are affected in Friedreich's. So this all comes from 1 3-millimeter piece of a skin biopsy from an arm of a patient. And as I mentioned, we have about 80 of those lines in our repository. And next slide shows you what we also do. We work with them in the lab, but we ship them. We ship them all over the world. Those cells are actually right now in India, in Australia, they are in France, in Sweden, in many other European countries. And on this map, you also see how many different cities in the United States have those cells. I think we made more than 100 shipments since we moved to UT Southwestern. Next slides actually show you how this entire process works. So this process takes about 4 months total, starting from this little piece of skin, which you see on the left corner. Then the next part of that slide is those skin fibroblasts grown, those skin cells. Then the next part is those round cells, which are induced pluripotent stem cells. And then on the right-hand side, you can see neuronal cells. On the left-hand side, if we could make a video, this actually is a video, you would see those are cardiac cells, which are beating. So this is part of our laboratory. I would say that about 20 or 30% of our effort goes to that repository. The rest goes to the basic projects in Friedreich's. And of course, this doesn't happen by me. I'm sitting in the office and organizing those things. What it happens thanks to is, on the next slide, is the people in the lab, Jill, Puri, and all other who are shown here, and all their projects. And the second thing, which is most important in our work, is the funding. This doesn't happen for free. We have funding from federal government, from Department of Defense, from National Institute of Health, but we have been blessed with the continuous funding from FARA. And thank you guys for that. That's what you contribute.